Now, humans have a very interesting pattern of genetic diversity globally. Whether from mitochondrial DNA, sex chromosomes, or autosomes, we find three persistent patterns that hold true for nearly every area of the genome. One pattern is that overall, the amount of genetic diversity for humans is much smaller than you'd expect with a species with 7 billion members. The second pattern focuses on alleles, those genetic types specifically. If an allele is common in one population, then it is typically common everywhere in the world. A third pattern is that the genetic variation of people outside of Sub-Saharan Africa is largely a subset of the variation you find within Sub-Saharan Africa. Let's talk about these patterns a little more. Earlier, we showed an ape phylogeny. This tree really brought home the point that even within subspecies of chimps, there's more nucleotide diversity than we see for the entire species of humans. In fact, while there are 7 billion people in the world, genetically together, our low level of genetic diversity looks like what you'd expect from, say, 20 to 30,000 people. Overall, genetically, humans are very, very similar to one another. The similarity of humans holds true even when we focus on alleles that are common to a population. For a specific population of interest, common alleles are typically defined as those alleles that are at least found in 5% of the population. One remarkable fact about humans is that if an allele is common in one population, then it is typically common everywhere in the world. For example, if an allele is common in one country like Japan, then that allele is also common in other countries like France and Iran and Peru, the US, everywhere in the world. Moreover, if an allele is only found in one region of the world, such as Western Europe, then very few people in Western Europe actually have that variant. There are very few exceptions to this pattern. That said, nucleotide diversity of human populations is not the same across the globe. There is greater nucleotide diversity in Africa than anywhere else. Here you can see the estimate of nucleotide diversity comparing individuals within and between Africa, Europe, and Asia. That study demonstrated that if we were to take two people randomly drawn from African continent, then on average, those two people would be more different from one another than one person drawn from Africa and one person drawn from anywhere else in the world. In other words, two people randomly drawn from Africa are more different than one person drawn from Europe and one person drawn from Africa, or one person drawn from Africa and one person drawn from Asia. Now, we've already mentioned that the genetic variation within Sub-Saharan Africa is really large. The genetic variation outside of Sub-Saharan Africa is largely a subset of that found within Sub-Saharan Africa. But the actual pattern is even more astonishing. For example, the genetic variation in Northern Africa is largely a subset of what you find in Sub-Saharan Africa. The genetic variation in the Middle East is largely a subset of what you find in North Africa. The genetic variation in Europe is largely what you find in the Middle East, as well as the rest of Central Asia is what you find in the Middle East. The genetic variation in Siberia is largely a subset of what you find in Central Asia. The genetic variation in North America is largely a subset of what you find in Siberia. And the genetic variation in South America is largely a subset of what you find in North America. So we're seeing this sequential series of subsetting of genetic variation, beginning in Africa and spreading to the rest of the world. Now, explaining this pattern of genetic variation is pretty easy if we revisit our lesson in population genetics. Please revisit that section because I want you to be able to understand this pattern of genetic variation, not as a story that I'm telling you, but rather as a logical deduction of how alleles are lost or perpetuated in populations over time. If you remember your population genetics, then it should be clear that the pattern of human genetic variation globally has largely been influenced by a sequential series of founder effects, beginning in Africa. Remember what happens with founder effects. With founder effects, the daughter population has a poor sample of the genetic variation of the parental population. Because of chance genetic drift, alleles that are common are more likely to be sampled than alleles that are rare. Thus, common alleles will tend to have a global distribution. This explains how an allele that is common in one area of the world will tend to be common everywhere else in the world.